Good morning, students. Today we will learn about translation, the process by which proteins are made in cells. Proteins are essential because they form structural parts of the cell, enzymes, hormones, and perform many vital functions. Let's start by understanding what translation means in simple words. It is the process of decoding the genetic information carried by mRNA to synthesize a specific sequence of amino acids, forming a protein. In this process, mRNA acts as a messenger, carrying genetic instructions from DNA in the nucleus to the ribosome in the cytoplasm. tRNA functions like a transporter, bringing specific amino acids to the ribosome according to the codons on mRNA. The ribosome is the actual site of protein synthesis, made up of large and small subunits. In the diagrams I'll draw, the ribosome has three key sites: the A site, P site, and E site. The small subunit of the ribosome initially binds to mRNA, and then the large subunit joins, forming the complete ribosome. Let's briefly understand the genetic code. Each 3-nucleotide sequence on mRNA is called a codon, which codes for an amino acid. The start codon, AUG, codes for methionine and signals the beginning of protein synthesis. Stop codons like UAA, UAG, and UGA do not code for any amino acid and signal the end of the process. Now we'll see how translation occurs step by step, starting with initiation. During initiation, the small ribosomal subunit binds to mRNA near the start codon, and the first tRNA carrying methionine attaches. In elongation, more tRNAs bring amino acids according to the codons on mRNA, and the polypeptide chain grows. The ribosome moves along the mRNA in the 5-3 direction, with the growing chain always adding amino acids at the C-terminal end. The tRNA first attaches at the A site, then moves to the P site, where the amino acid links to the chain, and finally exits from the E site. The chain elongates as amino acids are joined by peptide bonds, forming a growing polypeptide. Eventually, during termination, a stop codon appears, causing release factors to bind and releasing the completed protein. The entire process involves the translation of information from mRNA into a sequence of amino acids, forming a functional protein. Remember, the polypeptide chain grows from the N-terminal to the C-terminal, and translation always proceeds in the 5-3 direction. Understanding codons, tRNA, and the sites on the ribosome makes the concept of translation much clearer for NEET.